الحقيقة بقى يشرفني نعمل مسك الختام بتاع اليوم، وأنا اخترته مخصوص يبقى آخر توك لأنه حقيقة مسك الختام. أقدم أستاذ الدكتور إسماعيل رمضان، أستاذ النيورولوجي في جامعة اسكندرية. أنا مش هتكلم، هو يعني 3 سبيكر زي أنتوا هتلاحظوا، ده هيتكلم في موضوع ما حدش قبل كده بالنسبة لنا اتكلم فيه. بيتكلم على حاجة اسمها Central Insulin Resistance and its relation to neurological disorders. دكتور إسماعيل.
Thank you very much, Mr. Chairman. I'm really honored to be invited by Professor Khalifa few days, I think a couple of weeks ago, to do this lecture. This lecture, actually, I have 2 points I have to say before I present this lecture. First of all, most of the research or most of the papers that will be presented here are actually published in 2020 in the last 4 months. With the exception of just one that was published in 2014. Second, it's a very booming subject now on. Central insulin resistance and neurological disorders. This is actually my agenda. I'll talk about insulin and the brain, central insulin resistance, and its relation to some neurological disorders. First of all, in 1970, there was some kind of a myth that the brain was thought to be an insulin-independent structure, that they say that the brain does not depend on the insulin for the glucose metabolism or for the usage of the glucose. And this is actually logic, because the brain cannot actually be subjected to any changes in the insulin plasma level in order to be supplied with the glucose. So it is an insulin-independent structure. This was a myth. But the recent research showed that insulin receptors are abundant in the brain. We have a lot of insulin receptors in the brain. And they are expressed throughout the human brain on neurons and the glial cells, with actually some kind of a fluctuation in the concentration according to the area of the brain. That's to say, the cerebellum maybe has a lot of insulin receptors. The hypothalamus is a little bit less than the cerebellum, and the cortical and subcortical regions are actually having these insulin receptors, but in a less amount than the cerebellum and the hypothalamus. This is actually a diagram which shows the different concentration, where the cerebellum, the highest concentration, the hypothalamus, the cortex, the frontal cortex, the midbrain, the hippocampus, the nucleus accumbens, which, and the caudate and putamen and the amygdala, which are the nuclei actually responsible for the Parkinson's disease. But we'll talk about this later. So it was quickly appreciated that insulin signaling specifically modulates brain function with diverse metabolic or cognitive functions. So it has a different function in the brain, affecting memory, olfactory perception, emotional regulation, eating behavior, and peripheral metabolism. This is actually a different function for the insulin in the brain. Okay, now we have couple of questions to be answered. First of all, how insulin reaches the brain. Actually, this is a diagram how insulin reaches the brain. This is the blood and the plasma insulin level, and this is the blood-brain barrier, okay? And this is the brain. The insulin, which is present in the plasma, actually goes to be attached to certain insulin receptors here in the blood-brain barrier in order to be transmitted to the brain. And we have a nonlinear correlation between plasma and CSF levels of insulin. That's to say, it doesn't matter how much insulin here in the plasma, okay, because once the receptors has been saturated with the insulin, and it will be transported by the transcytosis, so no more insulin will go into the brain. So whenever these receptors will be saturated, okay, that's it. Insulin will not transmit to the brain. And we have a higher permeability at the pons medulla and the hypothalamus that actually transmit insulin from the blood to the brain at these certain areas, and lowest permeability at the occipital cortex and the thalamus. Okay. Now we have another question which actually pops a lot of ideas for the researchers. Could insulin be produced inside the brain? Most of the data that will be presented here are actually a research data, and there is nothing sure for these facts, but the facts are very solid, and they are going from one stage to another stage. First of all, the C-peptide was detected in the brain. We know that in the pancreas, pre-pro-insulin, part of it will be cut, then it will form pro-insulin, then part of it, which is the C-peptide, okay? They found this C-peptide in the brain. They found it, okay, which will actually, at the end, will form insulin. So this is one fact that indicates that insulin actually could be formed inside the brain. Not only this, but the insulin messenger RNA was detected in certain brain regions. In certain brain regions, they found the cells which are responsible about forming the insulin. Not only this, but actually they were released from choroid plexus epithelial cells and GABAergic interneurons. We know that the choroidal plexus actually are responsible for production of CSF. They are not responsible actually for the absorption of CSF. So when you find some kind of an insulin CSF, this might be produced by the choroid plexus inside the brain, which is another actually fact that insulin might be present or might be formed inside the brain. Please, I don't want you to be panicked by this slide. I will try to guide you through this slide. This is actually the neuron. This is the insulin in the interstitial fluid. So the insulin will actually go inside the neuron. This is the neuron. It has actually two main mechanisms or two main path of channels, two main ways inside the brain. One is what we call MAPK, and the other one which is PI3K, and this is the insulin receptors. One of the insulin receptors here is GLUT4, and this is just a receptor that will take the insulin inside the neuron to be acting on the glucose, okay? But the other receptors has totally a different function. When insulin goes inside the neurons, there are pathway. I just want you to remember this word, MAPK, one of the pathways, okay? And they will end with brain development and function and protection from degeneration. So insulin has totally a different function inside the brain. It's not related to glucose, with the exception with these receptors. It has a different function, which is brain development and function. Okay, this is Ras, maybe you are familiar with this. Okay, one of the molecular biology terms that actually very difficult to catch. But the main target from this slide that insulin has a different function inside the brain. This is the GLUT receptors, GLUT1 through to 3, 4, which are responsible about getting the insulin inside the brain to act on the glucose. This is the same diagram, with the exception is that there is another pathway which is responsible about the apoptosis and the oxidative stress. This is the insulin receptors, okay? So now the insulin have a function inside the brain concerning brain development and another pathway concerning with apoptosis. Whenever there's a pathway, okay, one of them is acting, the other one will be deactivated. And whenever this will be activated, the one which is responsible about the brain development and the brain function will be activated. So they are acting, okay, against each other. Okay, now how could we tell that there is an evidence of central insulin resistance. And I want you to pay attention to this because now I'm talking about some that might be facts about diabetes, okay, not neurological disorders. How did they figure out that there is what we call central insulin resistance? Okay, they transport some kind of intranasal insulin, okay? This is the experimental work, intranasal insulin, and they try to measure the areas in the brain which will be activated by this transnasal insulin. And they will measure this by something which we call functional MRI. Functional MRI is an MRI which actually measures the oxygen consumption of different areas of the brain whenever they will be activated. We'll have here red areas, okay? This means that this area is highly activated by the transnasal insulin, and bluish areas, which means that these areas are less activated. This is the normal, okay, for the intranasal insulin. Whenever they are delivered to the brain, these areas are activated. This is the normal persons. What they found out actually that this response is reduced or absent in individuals with obesity and or peripheral insulin resistance. Whenever you are having someone with peripheral insulin resistance, maybe with diabetes mellitus or something like this, or just obese, they found that this response is absent or reduced. So there's some kind of a relation between central insulin resistance and peripheral insulin resistance. And okay, let's now revise what the central insulin is doing, because it acts on the hypothalamus with modulation of peripheral glucose production. It acts on the hypothalamus with modulation of peripheral glucose metabolism. They are acting on the mesocorticolimbic circuit, which is actually in the prefrontal cortex and these areas, and has beneficial effects on eating behavior by reducing food craving. And the prefrontal cortex, when they act on these areas, actually it acts on the learning and memory, cognition, the human behavior regarding the motivation, reward center, and food intake and food choice. So the central insulin, some of the functions of the central insulin will, by a way or another, activate or deactivate the insulin peripherally, either directly or through modulation of certain functions. Does it start centrally or peripherally? This is the big question, actually, that they are searching for answers. The insulin resistance that we are looking for, the insulin resistance that causes diabetes, does it start centrally or peripherally? These are some interesting findings. I will just mention these findings, and I will let you think about it, okay? And maybe the near future will come up with another fact. First of all, intranasal insulin improved peripheral insulin sensitivity in men who are lean, irregular, or Rufaiyin. It doesn't have to do, it doesn't make any benefit, okay, with the females or with the obese. Maybe with the females, maybe not, because some kind of hormonal changes. Maybe the obesity because of the adipose tissue and the secretion of many hormones, maybe. But intranasal insulin improves peripheral insulin sensitivity in men who are lean. When they knock down insulin receptor in a specific region of the hypothalamus, actually this happened in rodents, known for glucose sensing, impaired insulin secretion, and resulted in glucose intolerance in rodents. What they do in this experiment, actually, that in rodents, okay, central insulin receptors, they knock it down, okay, and the results was glucose intolerance in rodents peripherally. And this actually document that there is some kind of relation between central insulin resistance and peripheral insulin resistance. Maybe it's not totally, of course, it's not totally dependent on the central insulin resistance, or the happening of the peripheral insulin resistance does not actually totally depend on the central insulin resistance, but definitely, according to these facts, it's one of the factors that might affect the occurrence of peripheral insulin resistance. Not only this, but there is an existence of brain-liver axis. Intranasal insulin administration improved hepatic energy metabolism and reduced fat content in lean and healthy people. Again, in lean and healthy people. Okay, now we have more data which is actually very interesting. Central insulin resistance can develop before birth. Impaired maternal metabolism due to obesity, associated insulin resistance, and gestational diabetes affected fetal brain activity and function of the autonomic nervous system. That's to say, we are having an obese female who is pregnant, giving birth to a child, and she has some kind of a problem that affects the central insulin in this fetus that was born with central insulin resistance, and later on he might develop diabetes. So what does this tell us? This tells us that it might be one of the theories of the reasons for occurrence of diabetes, that there is some kind of a central insulin resistance occurs in utero that will manifest itself later on with the occurrence of diabetes. Again, this is not the only reason that might be responsible for this, but definitely it's one of the reasons. Now, I'm going to talk about some of the occurrence of the central insulin resistance and some neurological disorders, okay? For example, in diabetes mellitus, we know that there are several risk factors, just like hypertension, smoking, head injury, high cholesterol level, and diabetes mellitus is one of the major risk factors for the occurrence of Alzheimer's disease. We know the pathology of Alzheimer's disease. We know that there is some kind of an accumulation of abnormal protein inside the brain. And we have drugs that actually will remove these proteins from the brain. But actually, we don't cure Alzheimer's disease. We only do some kind of a symptomatic treatment. So most probably, the pathological marker in Alzheimer's disease, which is accumulation of the protein, is not responsible for this disease. But we might have another reason that we didn't discover yet, okay, which is responsible about developing Alzheimer's disease. And a better understanding of the mechanism underlying the pathology is needed. Is there a role of insulin? There is bidirectional relationship between metabolic disease and cognitive function, because one of the major roles of the central insulin is concerning cognition. Diabetes type 1 and 2 increase the risk of dementia by about twofold, so definitely there is some kind of relation. Neurocognitive disorders have been linked to an increased risk of metabolic disease, so there is some kind of relation between cognition and diabetes mellitus. And there is hyperglycemia causing glucose neurotoxicity and accumulation of glycation in the products, plus the vascular injury, plus the dysregulated brain insulin, and this might be responsible. And recent research actually finding in Alzheimer's disease that insulin receptors, some of the insulin receptors that on the earlier slide were found inactivated. You know the pathway that I showed, responsible about the insulin, the MPK, and the RAS, and the mTOR, and things like this. And insulin binding affinity is reduced in these patients. So insulin desensitization is derived by chronic inflammation, which occurs in Alzheimer's disease by a tumor necrosis factor and the other inflammatory products. And these are, you know, this slide I showed earlier, and these are the things which are actually impaired in Alzheimer's disease concerning insulin. And there is some kind of therapeutic trials concerning Alzheimer's disease, several blind, blinded placebo trials where they improve recall of verbal memory and improve other cognitive function in Alzheimer's disease. And I heard a lot about the GLP-1, and maybe Professor Mim, she could take this slide, and it might have helped her with the debate against Dr. Nabil, but because this is one of the drugs that recently might be approved in Alzheimer's disease, so there is some kind of over diabetes. You can see Professor Nabil is highly advantaged. Peptides, okay, and this is a therapeutic trial. Vascular dementia, actually, they have the same, maybe several mechanisms and risk factors, small vessel dysfunction, hypertension, atherosclerosis, and diabetes mellitus, of course, is responsible for vascular dementia. And the insulin at high concentration binds to receptors in endothelial cells, which is the blood-brain barrier, stimulates production of endothelin, okay, one of the substances in the brain, blood-brain barrier, and causing vasoconstriction. And this is, you know, a diagram showing that actually insulin have a lot of deleterious effect on many of the neurological disorders. I won't go through this slide. And they are having actually the same pathophysiological mechanism concerning insulin and Parkinson's disease. Thank you very much.
أشكر حضرتك يا دكتور إسماعيل على المحاضرة اللي فعلا قيمة، وفعلا زي ما أستاذنا الدكتور خليفة قال كانت مسك الختام. أشكرك جدا يعني. هو بس بصفة حضرتك يعني قريت في الموضوع ده كتير، هل تعتقد إن إحنا لو استخدمنا الإنسولين سنسيتايزر TZDs يبقى له دور في البرين انسولين ريزيستنس لايك بريفرال انسولين ريزيستنس؟ في جروب اوف ميديكيشن فور تريتمنت اوف دايابيتس انسولين سنسيتايزر TZDs. هو المجموعه دي ريلي من اقوى المجموعات لانها تاتشنج واحد من الباثوفيسيولوجيكال ميكانيزم اوف دايابيتس which is insulin resistance. والحقيقه لما بنختار العيان كويس انه يكون انسولين ريزيستنس ويمشي على المجموعه دي بيفضل سنين طويله متظبط. هل تعتقد ان لها رول في البرين انسولين ريزيستنس؟
هو ديفينتلي الحضرتك بتقوله ده، يعني انا ما كنتش اعرف ان في حاجات من الانتي دايابيتك تريتمنت بتشتغل سنترالي، ولكن ديفينتلي لو في حاجه بتشتغل سنترالي وهاف مور beneficial effect than the other drugs which are actually acting peripherally. So definitely there is some kind of a central mechanism that is involved in the pathophysiology of diabetes, because actually we are having a lot of drugs, especially in Parkinson, in Alzheimer's, which are totally acting on the central insulin, and they have high beneficial effect on our neurological disorders concerning Alzheimer's and Parkinson mainly. So definitely أنا most of the data just بس علشان أقول اللي أنا قلتها هي اتقالت في اللانست اللي هي اتنشرت في آخر 4 5 6 شهور، لأن هم طلعوا كذا عدد concerning السبجكت ده ورا بعضيه في لانست. وأنا I have برضو لو حد عايز عندي الفايل كله يعني.
شكرا دكتور اسماعيل، احنا يعني استمتعنا بالمحاضرة.